Hello there. Thank you for joining me on this second lecture video, looking at the endocrine system. So this time we will be looking at some chemical aspects of hormones in a little more detail. How about, why don't we just get started right away? So hormones can be divided in two main classes. Firstly, amino acid-based ones. These are, for example, peptides and proteins. And the second group is steroids. These are all made of cholesterol. Remember from our nutritional talks in BIO201 and BIO156 that some amount of cholesterol is needed in body. But there is no need to get it from food. Your body makes all the cholesterol that is needed. And finally, I want to mention that this classification may very well get expanded in the future. Some scientists are considering eicosanoids as a hormone, while others still classify it as a paracrine. What will happen in the future? We will just have to wait and see. But I do want you to notice how the fields of science are constantly changing and how the concepts are being re-evaluated and revisited. This is what I find so exciting about working in this field. We are far from a finished product of fully understanding these concepts. Okay. Let's look at the actions of hormones in a little more detail. So, have you ever wondered of this? Since hormones are passed through the bloodstream to everywhere in the body, how come we do not see these hormones' effects and the changes that they cause everywhere in the body? Well, you might not have thought about it, but I certainly have. It is because only the cells with receptors for that hormone will be affected. These cells that have receptors for a particular hormone are known as target cells for that hormone. And what they do in the target cell, once they bind to the receptor, is that they affect the cell's activity in one way or another. So there must be a specific fit or a match, if you wish, between the hormone and its receptor. Only the ones that are an exact match will do. One way to think about this is like they would be a key and a lock pair. There are many hormones, just like there are many keys. And there are many different receptors, just like there are many different kinds of locks. But only a pair that is a match will bind correctly and alter the target cell's activity. So, what do I mean by it when I say that the hormones affect the activity of the target cell? Well, here I have a list of possible hormone activities. And as you can see, there are many. They can change what gets into the cell and what is blocked out. Or they might trigger or stop production of enzymes or proteins. They may also cause the cell to secrete something. Or they may even trigger cell division. So there are many possible effects on the target cells, and these are only some examples that I could think of. Another thing that I want to highlight is that the chemistry really matters here. How hormones act depends on their chemical nature and receptor location. And there are two main ways. Let's have a look at these. Just like you remember from our earlier discussion of vitamins in your early introductory biology classes, there are both water and lipid-soluble hormones. Let's start with water-soluble ones. These are typically amino acid based and act on the surface of the cell, on its membrane. So therefore, as the receptor is located on the cell membrane, additional extra steps are needed for them to do their job inside the cell. And these multi-step processes need a second messenger in the cell. Your textbook goes into quite a bit of great detail on this, but I think that we will just leave it to here for now. Lipid-soluble hormones instead are typically steroids and thyroid hormones. Because being lipid-soluble, they are now able to cross the cell membrane and enter into the cell. They act on a receptor that is located inside the cell. And their actions are much more direct. They act on the DNA and through controlling the protein production. So there is no need for these multiple extra steps like the water-soluble hormones had. So that's a rather nice comparison of the two, isn't it? I also want to talk a little bit about the release of hormones. This is a good time for us to remind ourselves about one of the very basic concepts that we have talked about in our previous courses. If I was teaching you at that time, I did tell you that it would be a fundamental one and we would come back to it often. And that is just what we will do here. And this was, of course, homeostasis. So homeostasis referred to the maintenance of constant internal conditions. And we had a rather nice simple diagram that I proposed to you. Remember, something changed. It was noticed by a sensory organ or a sensory structure. Message was then relayed, typically to the brain, where we had a range of acceptable conditions. And if the stimulus fell outside this acceptable set point that we had established, something needed to be done. And this something was done by an effector. And typically, this was to stop something from happening. And this was what we know as negative feedback. At the very end of this video, I have included a link to another lecture where we first discussed about homeostasis in our introductory biology courses, in case you have forgotten this concept. Remember that the negative feedback was more common. Here we stop a change from happening. You could say that we stop deviations from the set point. However, there was also positive feedback, which is much more rare, but can also sometimes happen. It takes the deviation from the set point even further. So we're going back to the release of hormones. Levels of hormones in the blood are constantly controlled by this negative feedback. And and they are maintained within a rather narrow window of the set point. This refers to what is an acceptable amount of hormone in the blood. If it deviates too far from that, the negative feedback control loop kicks in. Well, there are two ways how the hormone release can be triggered. Either by endocrine gland stimuli, which is hormone being released caused by an alteration in the level of iron or nutrients, for example. Or by a nervous system. This is nerve fibers stimulating the hormone release. Or it could be that the hormone release is caused by another hormone. This would be known as hormonal stimulus. I did not list it here separately. Finally, let's look at the target cells in a little more detail. These are very specific. Target cells must have specific receptors to which the hormone binds to. Remember our key and a lock mechanism, where they needed to be a match. So what may affect this? Well, I have 3 different points to highlight here. The amount of hormone in the blood, the amount of receptors in the target cell, and how well hormone and target receptor bind together. The amount of hormone that is present will also have an effect on the number of receptors in the target cell. If there are low levels of hormone around, the target cells make more receptors. This is known as upregulation. And if there are high hormone levels around, the target cells will lose receptors. This in turn is known as a downregulation. I guess that what I want to highlight here is as the most important take-home message that the human body is very adaptable, and it is the changes that we especially note, such as in this case, the changes in the amount of hormone around. The last 2 big questions that I guess that we should talk about here are, how do these hormones travel in the body? And what removes these hormones so that their levels do not just keep building up higher and higher? Let's have a look at these 2 questions. Hormones circulate in the blood in 2 possible forms, either as free or bound to a carrier or plasma. And this is why we do blood work on the patient. We get very useful direct information about the concentration of this particular hormone in the blood. And the concentration can be explained by 2 factors: how much or how fast the hormone is being released, or how much or how fast the hormone is being removed or inactivated. So I promised to look at also what removes hormone from the blood. Let's consider it. So hormones can be removed from blood by degrading enzymes, kidneys, or liver. Remember, both kidneys and liver have a main task of filtering the blood. And this gives us the concept of half-life. Half-life of a hormone is the time that it takes for a hormone concentration to decrease by half. And for some hormones, this may be just minutes, while for others, it can be weeks. So there's a lot of variation here from one hormone to another. And this depends on whether the hormone in question is water or lipid soluble. This is a rather nice table that I put together based on your textbook, summarizing the differences between water and lipid-soluble hormones. You can study it in more detail on your own time. Finally, multiple hormones can be acting on the same target cell at the same time. So let's look at what kind of an effect that would have. This can be either due to the hormone in question needing another hormone to be present so that it can have an effect. Or if another hormone is present, it may amplify the effect of the hormone that we are interested in. Or this other hormone may act against the hormone of our interest. So that's it. On this video, we have covered a lot. We have discussed the chemistry, actions, release, target cell specificity, and finally transport and removal processes of the hormones. I know that it has been a little theoretical, but as I said in our live online session last Wednesday, over the remainder of the course, we will refer back to the different aspects of endocrine system on multiple occasions. So endocrine system will become more familiar to you as we look at aspects of it as they relate to the very system that we are looking at that time. Thank you again for sticking with me for this video. I hope that it has helped you to clarify some of the important concepts of this chapter.